monthly Meet the Scientists webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. Bruce McEwen will present a webinar entitled Brain Plasticity. What is it and why is it important? The Brain and Behavior Research Foundation is dedicated to funding research around the world to identify the causes, improve treatments, and ultimately develop cures and preventative techniques for mental illness. The foundation has awarded over $300 million in research grants since we were founded a little over 25 years ago. I'm delighted to introduce Dr. Bruce McEwen. Dr. McEwen is the Alfred E. Mirsky Professor and head of the Harold and Margaret Millikan Hatch Laboratory of Neuroendocrinology at the Rockefeller University in New York. Dr. McEwen has written more than 1,000 scientific papers. He discovered that cells in the brain's hippocampus, a region important in memory and learning, had an abundance of receptors for stress hormones. The impact of this finding has proven to be enormous, completely changing understanding about the physiological impact of stress and how to treat stress-related conditions. In 1998, Dr. McEwen received NARSAD grant funding to support his quest to understand what happens when stress impacts and seems to damage the brain. While his research confirmed that stress does impact the brain and can cause shrinkage in the hippocampus region, for example, he also found that the impact is not necessarily permanent damage. He discovered the brain's inherent capacity to adapt and remodel its architecture, and his research effectively established what is known as neuroplasticity in the field. Dr. McEwen is a member of the Brain and Behavior Research Foundation Scientific Council that identifies the most promising ideas to fund with NARSAD grants each year. He is also the recipient of the Foundation's 2005 Goldman Rakik Prize for Outstanding Achievement in Cognitive Neuroscience. This will be an interactive event. We'll start with Dr. McEwen's presentation, followed by a question and answer period. To submit your questions, please use the Questions tab of the webinar control panel. You can submit your questions throughout the presentation. As your moderator, I will present your questions to Dr. McEwen, and we will address as many as possible. And now, it's my pleasure to present Dr. Bruce McEwen. Bruce, the stage is yours.
Thank you very much, Jeff. I'm delighted to be here, and I wanted to, before I started the presentation, I wanted to do a couple of quick polls. So you will be seeing these polls. Please fill them out, and then at the end we're going to do something to follow up and see how your views might have changed.
Okay, so the results are on the screen. 97% yes and 3% no.
Wow, okay. We do the next one?
Sure. A few more seconds and we'll close. Okay.
Okay.
And yes, so you can see the results.
Very good. Well, it'll be fun to see how things change as we go on, but you know, I can see I have some work cut out for me.
Okay.
All right. Okay, so what I'm going to tell you about really starts with the this next slide, which is that in the last 20 years we've realized that dendrites of brain cells change, can shrink or expand with experience, that synapses turn over continuously, at least a subset of synapses, and that there's neurogenesis, which continues in some brain areas and not especially in the hippocampal formation. And this is true for the adult and even aging brain, as well as the developing brain. So we've known for some decades that, for example, an enriched environment makes the cerebral cortex of a rat thicker. That we know now that regular moderate exercise increases plasticity, especially stimulates neurogenesis in the hippocampus. That intense learning is able to enlarge the hippocampus, not clear exactly what mechanism. That learning and practicing of motor skills alters neural architecture and circuitry. Likewise, sensory stimulation with vision or hearing, and I'll give some examples. And then I'm going to talk to you about acute and chronic stress, and also an important topic of sex differences and the role of sex hormones. So let's proceed. The hippocampus, as Jeff mentioned, is an area that we discovered somewhat serendipitously, has receptors for glucocorticoids, for cortisol. They are nuclear receptors that we originally discovered, although we hear that there are receptors for cortisol in other parts of cells that do other things. And we know that the hippocampus is important for memory, but also increasingly for mood regulation. It appears to be a target of depression, and we'll talk about that. Slides.
You can just click on the slide one more time.
Yeah. Okay. Okay, yep, all right. We're good. So when we think of stress, we think of cortisol. And cortisol, of course, is produced by the adrenal stimulation from ACTH from the pituitary gland, and that is stimulated by vasopressin and CRH2 peptide hormones from the hypothalamus. And their secretion is stimulated by both physical and psychological stressors impinging on the hypothalamus. We're going to talk about 3 areas of the brain that are targets in different ways for stress and stress hormones. The hippocampus, which beside its role in memory and mood, helps to shut off the neuroendocrine stress response. We'll also talk about the amygdala, which is important for anxiety, fear, and aggression. But it also helps to turn on, it does turn on the stress hormone response and increases heart rate. And the prefrontal cortex, which is important for self-regulatory behaviors, decision-making, working memory, mood, and impulse control, but it also helps to shut off the neuroendocrine stress response. Our original identification of the hippocampus as a target of stress hormones led us and others to look at this trisynaptic circuitry. You know, the information comes in from the entorhinal cortex. It results and it stimulates a reverberating circuit between the dentate gyrus and the CA3, and that in turn projects to the CA1, the trisynaptic circuit. And what we found is that repeated stress causes the apical dendrites of these CA3 neurons to become shorter and less branched. The basal dendrites don't change. And most importantly, this is not brain damage, because if you stop the stress, this apical dendrite shrinkage recovers in a matter of a week or so. Now, the mossy fiber terminals are from the dentate gyrus, release large amounts of glutamate, and I'll say more about glutamate in a minute. What we know is that the CA3 is very vulnerable to damage with seizures or head trauma. So one of the things that we suspect is that perhaps this is a protective mechanism, given the brain enough time to reduce the impact size of the excitation so it won't be so badly damaged. Now, the dentate gyrus, as you probably may know, it has neurogenesis. That neurogenesis is reduced by stress. I'll say more about that just in a moment. But first, talking about excitatory amino acids, we know that these mossy fiber terminals release glutamate. A study by Lowe, Galt, and Yamamoto in the 90s showed that during restraint stress, which is what we often use to cause dendritic shrinkage, that there's a peak of extracellular glutamate, so there's glutamate release. Interestingly enough, these workers found that adrenalectomized animals did not show that elevated glutamate. So it suggests the role of the adrenals. And indeed, we know that we can prevent the stress-induced remodeling, as we call it, by blocking glucocorticoid synthesis during stress, but also by blocking NMDA receptors, by using lithium and dilantin, which are stabilizing agents, by certain antidepressants, by benzodiazepines, and by a deficiency of the neurotrophin of BDNF. Now, in the dentate gyrus, as far as neurogenesis is concerned, we know that with repeated stress, and it took Cara Pham, a student at the time, for her thesis much longer than 3 weeks, which is our normal period to cause the dendritic shrinkage, up to 6 weeks, she could actually reduce the total number of granule cells in the hippocampus, reduce the amount of neurogenesis measured by the incorporation of BRDU, and in other words, cause that part of the hippocampus to become smaller. We know, and you'll hear momentarily, that neurogenesis is also stimulated by regular exercise and by an enriched environment. So the hippocampus has that capacity to go in both directions. Now, current studies just published in Molecular Psychiatry have looked at acute stress and acute novel stress for swim applied to naive animals, to animals that were stressed chronically for 21 days to cause that dendritic remodeling, or that they've had 21 days to recover after that chronic stress and were again challenged by that acute novel stressor. And it's very interesting what has happened. Look at the number of genes that are either increased or decreased. You see forced swim test compared to glucocorticoids, chronic stress, chronic recovery plus forced swim test. The most important point on this slide is that the immediate aftermath of chronic stress increases hugely the number of genes that are either turned on or off. And many of these genes are different under each of these conditions. So let's look further. What we've found in this study and some other studies is that stress activates a core set of genes, but not always in the same direction depending on the stress history. NF-kappa B system is one of them. CFOS is another. Chronic stress increases and changes qualitatively, which you just sort of saw hinted at on the previous slide, the gene response to an acute novel stressor. Recovery from chronic stress alters the pattern of gene expression. That is evident, and it's not the same as a naive animal given an acute novel stress. So even though dendrites recover, shrink, or grow back, it's not the same brain. Acute stress and glucocorticoid injections produce some but minimal overlap. Thus, stress is not just cortisol or corticosterone, and vice versa. That's important to remember. Acute stress activates a protective response to repress potentially disruptive genetic elements. I'll tell you about that just momentarily. And individuals with similar genomes differ in their vulnerability to stressors due perhaps to early life events. We can see this in terms of certain gene responses which predict their vulnerability to stressors. And finally, epigenetic factors, probably early in life, but throughout life appear to be involved, and I'll say more about that shortly. Several concepts that are important to know about is that there are what are referred to as context-sensitive genotypes, and maybe individual genes like the serotonin transporter alleles and the BDNF alleles. And in the ones that are more context sensitive, referred to as orchids, because orchids are very sensitive to the environmental conditions, the individual does worse in an adverse environment, but better than average in a positive environment, compared to dandelions, who of course can grow in many environments. And since we're about having spring upon us, we're going to see a lot of dandelions in the northeast U.S. Then also non-shared experiences, very important, even among individuals who are genetically identical, like identical twins. And we'll talk about epigenetic factors, including DNA methylation, that are very important. So as you probably know by now, epigenetics, which simply means above the genome, refers to gene-environment interactions that bring about the phenotype of an individual. This includes modification of histones, which I'll talk about in a minute, binding of transcription factors to DNA, response elements, methylation of cytosine bases without changing the genetic code, and microRNAs and various non-coding RNAs that we are beginning to understand some of their regulatory functions, but obviously it's very complex and very early in the game. If you're interested, you can look at PBS, Public Broadcasting System, under NOVA Science Series, and put in epigenetics. You'll find a 13-minute video which is very good in explaining some of this. Histones are involved in controlling the repression or expansion of the DNA double helix. If it expands, it opens it up to be accessed by transcription factors and modification enzymes and so forth. And if it's repressed and contracted, then those genes are inaccessible. And I want to give you one example of this from some recent work. Dr. Richard Hunter, a former postdoc now in Boston, showed that a repressive mark, this is histone 3 lysine 9, which is trimethylated. Now, it's not just cytosines that are methylated, but histones can be methylated. And you can see here on the right that an acute restraint stress of a naive animal causes a huge increase in intensity of this immunostaining in the dentate gyrus, and also a significant increase in parts of the Ammon's horn. Now, what does this mean? When he used a chromosome immunoprecipitation method to precipitate the DNA, find out what was repressed and trapped, he found that it contained a number of so-called retrotransposon loci, which are pieces of DNA that used to be called junk DNA, but now we recognize they contribute, among other things, to chromosomal rearrangements and other aspects that may be good or bad for cell function. And that repression, he showed, reduced the non-coding RNA that's transcribed from those loci. So this is important information, particularly since he found that repeated stress caused this repression to go away, and apparently aging shows this. So I'll say more about that just in a moment, but let's just remind you that as part of epigenetics, there is methylation and in some cases demethylation of cytosine residues, CpG cytosine near guanine residues in the DNA, and this is an important process that I don't have time to talk about today, but it goes hand in hand with the modifications of the histones. So getting back to this inverted U-shaped dose response curve where glucocorticoids facilitate excitation at moderate physiologic doses, they improve excitability, improve certain aspects of memory, so you remember things that are going to be important for your survival in the future. And then a more intense acute stress actually can begin to suppress excitability and suppress other kinds of memory. There may be a dose sensitivity of different classes of memory depending on how important they are for survival. And then there is the adaptive plasticity, which I have just been talking to you about, involving dendritic remodeling and neurogenesis. There is then the extreme of this when the brain is traumatized by a stroke, seizure, or head trauma, as Robert Sapolsky showed. Then the glucocorticoids and excitatory amino acids synergize and cause permanent irreversible cell loss. Now there's another state, which is loss of resilience related to aging. I'll give you an example shortly, but also probably in mental health disorders such as depression and anxiety. That is, a response with anxiety and maybe even depression is appropriate when a major life event or a very threatening event has occurred. And so the individual either retreats, hides away, becomes more vigilant in the case of acute threat, and that's fine. But if after the danger has passed or the recovery and exposure to a better environment, if those brain changes don't reverse, then you have an anxiety or mood disorder that has to be treated with some external means, and we'll talk about that as well shortly. There's been a lot of translation by other laboratories of this work on the rodent hippocampus to the human hippocampus, and we know that the human hippocampus shrinks over years with prolonged major depression, that it is smaller in type 2 diabetes, and that insulin and insulin resistance is a very important factor to be considered along with glucocorticoids and excitatory amino acids. And in fact, type 2 diabetes impairs mood, memory, and maybe a risk factor for Alzheimer's disease later on. Post-traumatic stress disorder involves perhaps a smaller hippocampus to begin with, but also some evidence that it shrinks further. So the smaller hippocampus, as a result of perhaps early life events, may be a risk factor of PTSD when something traumatic happens. Cushing's disease, of course, with elevated cortisol. There's some evidence that if you successfully reduce the cortisol by surgery, then mood impairment, memory impairment are somewhat reversed, and hippocampal volume, which is reduced, increases at least partially back to pre-Cushing's levels. But you don't have to have a disease to see hippocampal volume changes. In chronic stress over a 20-year period, there's smaller hippocampus. In chronic jet lag, people who travel like air crews without sufficient time to recover between international flights will show a smaller hippocampus. Lack of exercise, I'll say more about that in a moment, and chronic inflammation, which is often associated with things like diabetes, arthritis. It's part of the aging process, may be associated with a smaller, is associated with a smaller hippocampus in some studies. The good news is that hippocampal size increases with regular exercise, with intense learning, a study in German medical students, and with antidepressant treatments, remembering very few studies with the chemical antidepressants, but regular exercise is one of the best antidepressants. And to show you this, there's a study by Erickson and colleagues, Art Kramer at the University of Illinois, a number of years ago, showing that if elderly couch potatoes get off their duffs and walk around a running track for an hour every day, 5 out of 7 days a week, that over a period of 6 months to a year, both the left and the right hippocampus get larger, and their memory improves. It also happens their executive function and their prefrontal cortex is also improved. So we'll come back to the prefrontal cortex momentarily, but this shows you the power of a behavioral intervention to actually change the brain. Now, I've talked about how neurons in the hippocampus shrink with repeated stress. This is also true of the medial prefrontal cortex. But under the same conditions of chronic stress, while these neurons are shrinking, there are neurons in the basolateral amygdala and in the orbital frontal cortex that are expanding. The amygdala story is the work of Sumantra Shona Chatterjee in Bangalore, India, who's been a pioneer in this area, and you'll hear more momentarily about that. In fact, studies on the amygdala with both imaging in humans and studies in animals show that the amygdala is overactive in anxiety and depression. The children of long-term depressed mothers, work of Sonia Lupian in Montreal, have an enlarged amygdala, that is, living with a depressed mother who is not successfully treated over a 10-year period. The kids actually have an enlarged amygdala and presumably anxiety and proneness for depression. That effective treatment of chronic stress and anxiety can reduce amygdala volume. There's a study that a mindfulness-based stress reduction program, if it reduces anxiety and stress perceptions, will actually cause the amygdala to decrease. It's only one study. We need more of them like this. And finally, studies in the realm of cardiovascular disease show that an overactive amygdala and an underactive prefrontal cortex, which normally inhibits the amygdala, is a risk factor for early signs of cardiovascular disease. And then finally, timed glucocorticoid elevation appears to reduce PTSD symptoms. This is important because one's impression is normally that cortisol is bad, always bad in terms of these conditions. But in this study, which started with some epidemiologic work where people prone for PTSD or with PTSD had low cortisol levels, a study by Gustav Schelling in Germany showing that giving supplemental cort during open heart surgery for people with low normal cortisol levels, not Addisonian, actually reduced later PTSD symptoms. And then an animal model that we collaborated on with Shona Chatterjee and another animal model by Huggy Cohen and colleagues showed that that a timed elevation of glucocorticoids before or at the time of the trauma would actually prevent or reduce the development of PTSD symptoms. And in this paper noted here, there's a study of taking people after a traffic accident into an emergency clinic and giving them supplemental glucocorticoids, showing that they were less likely to develop PTSD. So here's another example of protective aspects of glucocorticoids, along with their aspects of memory enhancement. And one more thing I didn't mention, which is that acutely glucocorticoids and adrenaline actually enhance adaptive immunity. So there are a lot of things that are happening that are very positive, as well as the negative aspects that we're well aware of. Now, when we're talking about the hippocampus and amygdala, which communicate, and the amygdala also communicates with the prefrontal cortex. The prefrontal cortex is extraordinarily important, and we've been fortunate to work collaboratively with John Morrison at Mount Sinai School of Medicine, who's an expert in this area. And I'll show you just a glimpse of some of the results. Again, the removal or shrinkage of dendrites in the medial prefrontal cortex. This is an age-related study. This is what happens with young 3-month-old rats when you chronically stress them the way I've been talking about. They will show amygdala hypertrophy and retraction of dendrites in the hippocampus. They also show retraction of dendrites and reduced branching as well as length after 21 days of chronic restraint. And if the restraint, the stress is stopped, then over 3 weeks they recover. But I should add that the dendrites that recover, even though their length and branching looks the same and there are neurochemical features that are recovered, they are different dendrites because they are more proximal to the cell body than the ones which shrank to begin with. Again, reinforcing the idea that the brain is continually changing, and that even if it's resilient to an acute stress, it's a different brain. Now, in a middle-aged 12-month-old rat, living in ordinary laboratory conditions, not in an enriched environment and so on, but living 2 to 3 per cage, you see a retraction of dendrites, debranching, very much like you see in the younger animal, but given 3 weeks, there's only partial recovery. And then the old guys, 20 months old, you see the same degree of retraction, but no evident recovery in those 3 weeks of recovery period. Now, that doesn't mean we couldn't make this recovery with some experimental manipulations like an enriched environment or perhaps certain pharmacologic interventions. We just don't know that yet, perhaps even a better diet. This is work, research for the future. Another factor that's very important from work of Dr. Ilya Karatsorios when he was a postdoc, now he's at Washington State University, is that circadian disruption, which is sort of like shift work. What he did was to create a 10-10 light-dark cycle. Now, you know, we normally have a 12-12 light-dark cycle, and we have a clock in our suprachiasmatic nucleus that helps us to adjust to that 12-12 roughly circadian cycle. Well, in a 10-10 light-dark cycle, the brain, the clock does not adapt, and so there's a continuous sort of free running sort of or tracking of this clock, so the body temperatures at different times of day, and what happens over several months, even a month, is that the animals become heavier. They have higher insulin levels and higher leptin levels, suggesting that they're both leptin and insulin resistant. And we do know that the brain has receptors for leptin and for insulin, as well as IGF-1, and that they have important effects on the brain. And that neurons in the medial prefrontal cortex, like after restraint stress, become less complex, and the animal shows cognitive inflexibility. Is less able to shift, which is a characteristic of an atrophied or somewhat impaired prefrontal cortex. Now, a student, former student, Connor Liston, who did some of the basic work on the rat prefrontal cortex and effects of stress, teamed up with B.J. Casey across the street at Weill College of Medicine, and with us, took medical students studying for the board exam and going through their normal stressors of a year of medical school, used a perceived stress scale, which measures how much in control or lack of control of your life you feel. They put these people, volunteers, into an imaging machine, an fMRI machine, and gave them a task of cognitive flexibility. And then they also were able to look at a circuit that was activated by that task, and they looked at functional connectivity, which is the only way right now we can see if the circuits in those areas of the brain are impaired. And what they found, as you can see here, is that one of the parts of the circuits was the prefrontal cortex. Higher scores on the perceived stress scale predicted poor mental flexibility. They also produced reduced functional connectivity. But the good news is that the effects wore off after a vacation. So the brain, at least of the younger medical students, is resilient, like the brains of the younger rats. And the question is for older people and older animals if one can intervene, because we know, as I told you earlier, that a regular exercise regimen actually will increase neurogenesis and increase the volume of the hippocampus. So there is considerable hope. Now, everything I've talked about as far as animal experiments is based on male rats. If you subject female rats with their ovaries intact to the same kind of stress Well, in first place, we know that in males, if you do retrograde tracing, put a dye into the amygdala, have it trace back to the prefrontal cortex, that some neurons project to the amygdala, others project to other cortical regions. And it turns out that in the males, the dendrites that shrink with chronic stress are ones that project to other cortical neurons, not to the amygdala. But in females, these neurons do not shrink under chronic stress, but the ones that project to the amygdala actually expand their dendrites. And this only happens when their ovaries are intact or after we've given them estrogens. So, among other things, we suspect there are many other subtle sex differences that remain to be discovered, but just a little more about this, this sex difference. In a recent review article in Neuron with John Morrison, and based upon this study by Rebecca Shansky, here shows that in the presence of estrogens, the neurons projecting to the amygdala of the female rat expand their dendrites, and these are dendrites more proximal to the cell body. In the absence of estrogens, as after ovariectomy, chronic stress has no effect. Now, in the female, even though there is not an effect on dendritic shrinkage of the neurons that are projecting cortically, stress does cause an increase in spines, but estrogens inhibit that increase in spines in the cortically projecting neurons. And in the amygdala projecting neurons, there's an increase in spine density in both cases, whether there's estrogens on board or not, as a result of chronic stress. So it's a very complicated story, but it certainly suggests there are a lot more subtle sex differences in the brain, and we have to take that in consideration in trying to translate what we do in male rats to females, and let alone thinking about what that means for human beings. Now, the mechanism for this spine induction by estrogens in the prefrontal cortex, and also what I haven't told you yet, that occurs in the hippocampus of a cycling female rat, is a non-genomic effect involving receptors that are in spines, in the spine apparatus, in mitochondria, and certain synaptic terminals in glial cells. These are receptors that were discovered by, at the electron microscopic level by Terry Milner, a colleague at Weill College of Medicine, as more or less a serendipitous finding. And we now know that all classes of steroid receptors have forms, some of them alterations of the same genetic product, that exist near membranes and that have other functions, signaling functions, inside cells. It's not just the nuclear actions. And this is made even clearer for glucocorticoids if we look at this slide, showing that besides the nuclear effects that we originally identified when we found the hippocampus was a target for adrenal steroids, that there are indirect effects stimulating receptors that have actually tether together with other transcription factors to affect gene expression. But that's not all. Glucocorticoid receptors translocate into mitochondria, and at physiologic levels they buffer the mitochondria and help them take up calcium and reduce the free radical load. But at higher levels, as Robert Sapolsky showed, there is what he called glucocorticoid endangerment, and this mechanism fails, and there's increased free radicals and cell destruction because of over-excitation. But that's not all either, because glucocorticoids work directly on mineralocorticoid and possibly also glucocorticoid receptors that are membrane associated, and they help stimulate glutamate release, but at the same time they stimulate through a membrane associated receptor of the production of endocannabinoids, which are lipid products, the reason that marijuana is effective in the brain. These bind with a CB1 receptor on the presynaptic terminal, And the consequence of this is the inhibition of glutamate release. Now, in other cells that are specialized for GABA, the same mechanism can inhibit GABA release. So there's a complex mechanisms of counter-regulation, regulation, and counter-regulation, especially related to excitation, that make the glucocorticoid story, and by that parallel studies on estrogens, much, much more complex, but much, much more interesting for basic neurobiology. And these are receptors for glucocorticoid receptors with two different antibodies to the nuclear type receptor seen in postsynaptic densities, and one can see them in other parts of the cell. So this seems to be a real phenomenon for all classes of steroid receptors and virtually also throughout the body. So although I've hinted at this, there are multiple mediators of structural plasticity and epigenetic change. It's not just the steroid hormone working alone, and I've emphasized that time and time again. Stress is not glucocorticoids. Glucocorticoids are not stress. When there's stress, glucocorticoids work synergistically in a milieu that is also influenced by other hormones and by the sex of the animal. So sex hormones regulate spine synapse formation and permissive effects on dendritic growth. IGF-1 is involved. It's a liver hormone that's involved and it's essential for exercise-stimulated neurogenesis in the dentate gyrus. That the FGF family and BDNF are facilitators of plasticity both during development and in adult life. The corticotropin releasing factor is found in other neurons throughout the brain. Tissue plasminogen activator and lipocalin all mediate stress effects on the hippocampus and amygdala. CRF seems particularly important from the work of Tali Barham's group at Irvine. So they're also involved, and in a cell and region-specific matter. Endocannabinoids are important. The lack of the CB1 receptor leads the animal to be much more anxious and much more strongly affected by stress in terms of dendritic changes and synaptic turnover. And then I've also mentioned leptin, insulin, ghrelin, as well as IGF-1 as hormones that have direct effects on the brain by being transported into the brain. So what these stories have begun to teach us: hormone actions co-opt and participate in multiple mechanisms from the epigenome to rapid signaling and participate in adaptive plasticity. That structural plasticity is not necessarily damage and is reversible up to a point that changes with age. Yet the resilient brain has different gene expression responses during and after recovery. Early life experiences I haven't talked about do something called biological embedding, which is reflected in brain structure and function and in the propensity of the brain to respond in the ways I've described. That stress effects involve more than glucocorticoids, and that there are sex differences in brain structure and function. That may be much more widespread than we have expected. So what can we do about interventions besides using pharmacologic agents? I've already talked about regular physical activity. I've mentioned the mindfulness-based stress reduction that changes amygdala volume. And there's also a study on social support and integration, the Experience Corps getting elderly volunteers to help become teachers in an elementary school. They feel meaning and purpose in life, and I think we underestimate the importance of that, especially for elderly people, but for all of us. And there's something called eudaimonia, which is really reflects on this, this feeling of having some meaning and purpose in what you're doing. Another aspect of this is that we tend to regard antidepressant drugs, for example, psychotropic drugs, as like penicillin, that they'll cure a disease. Well, we know that's absolutely not true. And with respect to SSRIs like fluoxetine, there's an important study by an Italian group showing that if you treat animals with amblyostomia, that is unilateral or monocular vision because one eye was closed at birth, treat them with fluoxetine while stimulating the visual system, this will restore binocular vision to a large degree. A group in France took this same trick and gave fluoxetine to people recovering from a stroke, doing physical therapy, and showed a significant acceleration of recovery. And there's work by Errol Castrain and colleagues that, again, reinforces the idea that a drug like fluoxetine, together with a targeted behavioral intervention, is needed for example, treatment of depression. The same Italian group came back and said, well, there's nothing magical about fluoxetine because we can do every other day caloric restriction and stimulate and see the same results, restoration of binocular vision. They showed in the same paper that you can put corticosteroids into the drinking water instead of food restriction or fluoxetine treatment and stimulate the visual system and get recovery. Now, this brings up the final point, which has to do with the fact that there's naturally an ultradian pulsatility of our glucocorticoids that's very important for function. And Connor Liston, whose name I mentioned earlier, showed that in a thin window preparation where you can visualize spines forming over 24 hours or disappearing over 24 hours, that a tiny dose of dexamethasone, which doesn't get into the brain, it's pumped out, it shuts off ACTH secretion and creates a chemical adrenalectomy. The brain is not exposed to its own glucocorticoids. This spine turnover stops and can be restored by giving back glucocorticoids. And in a more recent paper, he's shown that the circadian variation of this pulsatility is very important for, among other things, for motor learning. And so what I think we have to think of is that although we tend to think of glucocorticoids as heavy-handed tools for suppressing inflammation and maybe also a part of bad stress, as people call it, that in fact, given a chance, the body has considerable wisdom. This pulsatility is part of normal processes that maintain plasticity, and this suggests that we should think about our lifestyles and make it more possible for the body to help itself. So looking to the future, we're talking then about plasticity, and we're beginning to recognize its potential. No, it's hard to say where this is going to go, but it's certainly an exciting frontier. These are the wonderful people that have been involved, and some of them involved in this study. And I think that's the end of my presentation. And now I think we're going to have another little poll.
We'll just leave a few more seconds, and then we'll close and share the results.
Good. Well, I think you've gotten many of the messages, and now I'd be very happy to answer questions.
Great. Well, first of all, Bruce, thank you very much for a presentation filled with information. And I think that although a very complicated subject, you were able to explain it in a way that I think had an impact on people's understanding of the subject. So thank you for that.
Thank you.
Yeah. The first question goes back a little bit to lifestyle, and in terms of at this point in time, if we're giving advice to people, how much exercise should people do? What would be the amount that you would suggest to somebody to have as healthy of a brain as possible?
Very good question. I think one does not have to become a marathon runner. That regular physical activity during the day, not just in the morning or the evening, but moving around from your desk or whatever you're doing on a regular basis is very important. There's something called NEAT, non-exercise activated thermogenesis, which is fidgeting. People who fidget are less likely to put on weight than people who don't fidget. So regular moderate exercise is probably the single most important thing a person can do, along with getting good sleep and also having a good group of friends and being sort of not being isolated.
When it comes to the sleep component, what would be the ideal range of sleep that a person would have?
Well, I think that's generally quoted about 6 to 8 hours. But excessive sleep may be a sign of some psychopathology, and too little. I mean, we know that shift workers are the ones that really get hit because they're trying to keep track of their daily lives with their families, and yet they're having to shift periodically to a new work schedule. And those of us who go through life with long commuting hours and so on may find themselves trying to have recovery sleep on weekends, and this works up to a certain point, but isn't totally effective, especially as you get older.
And can I ask you, above and beyond exercise and sleep, any suggestions in terms of proper diet and eating habits?
Well, I think obviously it's having some control over the amount of food, total amount of food one takes in. We all do this. People who indulge in comfort foods and so on, understandably sometimes because they're stressed out, maybe doing themselves a disservice if this is excessive. The other, of course, I'm not. Other people have popularized this caloric restriction every other day. It seems to work for some people, but not everybody. Probably better to do caloric restriction than to do total starvation or the yo-yo dieting, which some people do, which is very bad. But having a balanced diet that we all hear about and perhaps try to maintain.
So I remember when we spoke once, when I had the pleasure of interviewing you for the Healthy Minds TV show, that ultimately a lot of the science is demonstrating what our grandmothers all told us, which is proper sleep, proper exercise, and proper diet are good for you.
You got it. I mean, that really is the bottom line. But now, because we're understanding more of the mechanisms, and particularly important, I mean, for example, the recognition of the connection between a condition like diabetes and the brain, that we really understand the ways that the brain and the body can help each other, but also can harm each other.
Right, right. I want to talk a little bit about post-traumatic stress disorder, because it's been something that people are concerned about, especially in the context of our returning service members who've served our country, and some of whom have now developed post-traumatic stress. What does the current research say about what can be done to improve treatment for these people?
Well, that's a very good, very puzzling but important question. I mean, I think, and of course not just our returning service people, but people in countries like Syria and so on, where they have had, such conflict, war, and so on. I think that there's a big debate, as you know, between the so-called exposure therapy or repression, the idea of reactivation of memories. You can, for somebody who's been traumatized, and then has had many experiences over time, not just a single traumatic event, it's very hard to wipe out all those memories. But by reactivating some of them, since these fear memories never really disappear from the brain as far as we can tell, at least so far, they can be redirected to things that are more positive and perhaps less threatening. And so I think that's one of the ways. And there are some, pharmacologic agents that may be useful in that regard. But I think the general idea of opening windows of brain plasticity through the sort of reconsolidation mechanism and then letting the reassociation, letting behavior take its course is the best way to think about PTSD treatment, just as it is, I think, for depression.
And the issue of neurogenesis, I've often said that when I went to medical school, I was taught that old brains don't grow new cells, and old meant after the age of 2 or 3.
Right.
We now know that statement is incorrect, that
Indeed.
adult brains grow new cells. What does that mean for people? What's the take-home message about the fact that a person's brain can continually grow new cells?
Well, I think the example that I mentioned of the 65 to 70-year-old volunteers in Illinois, couch potatoes, walking an hour a day, 5 out of 7 days a week, and actually enlarging their hippocampus, is most likely explained by a stimulation of neurogenesis, perhaps also by a growth of dendrites and so on, showing that it is possible to make a difference no matter when in the lifestyle. And of course, we suspect that diet may play a major role in facilitating this, having omega-3s and doing the good stuff that may also make a positive difference. And in this concept of sort of eudaimonia, doing things that have meaning and purpose are very important. So I think all of these things may very well are probably reactivating neuroplasticity, not just neurogenesis, but the ability of other neurons to form new connections. And of course, we're still at the very early stages of having documented evidence for a lot of these things. Some of these brain game things on maybe making over too many claims about what they may do, but there is this plasticity, and neurogenesis is certainly the one that catches people's attention, for rightly so, but there are other forms of plasticity that go hand in hand.
So that exercise, which can promote neurogenesis and neuroplasticity, above and beyond what people have thought about as the endorphins that occur when somebody might go out jogging, there's the added effect of really growing new cells and making new connections in the brain that exercise brings about.
Exactly, and there are studies that show if you push it too far, that is, exercise too intensively, it can actually inhibit neurogenesis. So a person becoming fit means that they're gradually increasing their capacity for activity, and that is associated with these positive effects. If you push it too far too fast, you're going to have negative effects, and it may be deleterious.
We could go on for another hour on this, Bruce, and I'm going to want to have you back at some point in the not-too-distant future. This has been an extraordinary presentation and conversation. I want to thank you so much for joining us, and more importantly, thank you for the research that you've done that has brought about a real change in our understanding of the brain, and that I think will continue to have an impact on our understanding of the brain.
Thank you so much. I really have appreciated having this opportunity and being able to participate in this conversation.
Great, thank you.
Yep.
I also want to thank our audience for joining us today. The Foundation, through our research grants, is dedicated to to improving the lives of people with a wide variety of psychiatric conditions. And I want to thank people for joining us on that mission. All of the research that we fund is made possible through private donations. So if you'd like to make a gift, please visit our website, bbrfoundation.org, or call 1-800-829-8289. This webinar has been recorded, so if you've missed any portion of the presentation or would like to share it with a family or friend, people can visit the webinar page on our website. And I want to invite people to join us again next month as we'll hear about a different topic from Dr. John Rubenstein, the Nina Ireland Distinguished Professor in Child Psychiatry at the University of California in San Francisco. He will speak to us about understanding brain development as a foundation for understanding brain diseases. And that will take place on Tuesday, May 13th at 2 o'clock Eastern Time. With that, I'm going to again say thank you all and enjoy the rest of the day. Take care. Bye.